Okay, thank you, Christian. I am Erlend Sørmo, and in my talk, we'll be moving away from agriculture and related topics and onto waste handling and sorbents, of course, related to pyrolysis and biochar still. So I work at the Norwegian Geotechnical Institute, which is an institute for geotechnics, as the name kind of suggests, but also for environmental engineering and the kind of a combination of the two. And I would like to acknowledge some of my co-authors before I start. So Gabriela Castro at the Norwegian University of Science and Technology, who did a lot of the analytical PFAS work in this talk, and Katinka Kran, who was my master's student last year at the Norwegian University of Life Science. She's now working at the Lindum Waste Handling Company. She's also involved in this project. And my two supervisors, Hans-Peter Arp and Gerard Cornelissen, both working at NGI, but also have positions at the Norwegian University of Science and Tech and Life Sciences. So what I'll be presenting today is data from two research projects called VOW and sludge effect, and they deal with pyrolysis of contaminated wastes. So with a big focus on sewage sludge. And yeah, before we continue, I'll probably say that Scanship also needs a mention here. They provided a technology, the pyrolysis technology, and Bea's wastewater treatment plant. So before I get into the actual studies we did, I just want to add some background. So, I guess a lot of people have maybe a background in agriculture here, but we are now looking into pyrolysis as a waste treatment alternative for contaminated wastes. And some recent studies have shown that pyrolysis is a nice way to treat contaminated sewage sludge. Then, of course, the focus is to remove a lot of the contaminants that we have realized are in sewage sludge: organic contaminants, heavy metals, pathogens, yeah, pharmaceuticals, antibiotics, and a lot of weird stuff. Microplastics, of course, let's not forget. And the thing is that if we compare pyrolysis of sewage sludge to incineration of sewage sludge, then pyrolysis comes out on top because we get carbon capture and we get a biochar product that we can use for something. And this was very nicely demonstrated by Barry et al. In this 2019 paper. They did an LCA comparing sewage sludge pyrolysis to incineration, and looking at this global warming potential. Biochar route comes out a lot better than incineration. There are also some very recent studies that show that in the pyrolysis process, a lot of tricky organic contaminants are destroyed or decomposed, like pharmaceuticals and PCBs, PHs, endocrine disrupting compounds, to mention a few. And also this recent study from our group on organophosphorus flame retardant, and that's OPFRs, which will be published soon. But then the question is, how does pyrolysis work as a thermal treatment for sewage sludge that contains a lot of PFAS? And why do we wonder about that? Well, I'll tell you what PFAS is before we move on. They are a group of very persistent chemicals. They have high thermal and environmental persistence. They have gotten this fancy nickname forever chemicals because they're hard to break down. And the reason they're so hard to break down is because they have If we look on the right side here, they have this, let me just get the laser pointer here. Usually they contain this chain of carbons that are saturated with fluorine. And this carbon-fluorine bond is very strong. It's the strongest in organic chemistry, so it takes a lot of energy to break it. And these chemicals are also quite mobile, so they get around, usually end up in our water supply somehow, and they have been linked to a lot of different toxic properties like carcinogenic properties and hormone disrupting properties. And they are used in a lot of different applications, like really a lot. And there are thousands of different PFAS molecules that have similar structures with small variations, like a different functional group or a different length of this chain with some branching or whatnot. So we find them in firefighting foam, in your textiles, like waterproof textiles, like Gore-Tex clothing, and non-stick surface coatings, like your Teflon frying pan or paper cups and plates, also in personal care products, detergents. They're used in electroplating and building materials and much more. So they pretty much end up everywhere. They are found in rain all over the world and in our bloodstream, like almost every human. And sewage sludge is also a sink for these chemicals. It's not so strange because they come from all these different sources and follow our wastewater. Now, before we get on to the actual study, I just want to mention briefly also what a sorbent is, because some of you might not be familiar with this term. So a sorbent is a material that binds a contaminant strongly. Examples are ion exchange resins, fibers of different types, or activated carbon, and of course, biochar. And the biochar used for sorbents is considered fairly high value use. So you can use them in wastewater treatment plants, landfills, industrial air filters, and for remediation of soil and sediment. And type of application, like here in the wastewater treatment plant, is typically you have a slurry with activated carbon. And then you pump this dirty water in. As it rises through the slurry, the chemicals are sorbed into the pores of the activated carbon, and the water that comes out on top is clean. All right, so on to our research questions. As my title suggested, this was twofold. So the first part is what happens to these PFAS compounds that are found in the sewage sludge when we pyrolyze the sludge and produce biochar. Our hypothesis was that given a high enough temperature, they will be decomposed, so we remove them. And then can we take this biochar product and use it to sorb PFAS in, for example, wastewater treatment? Because we know already that some biochars actually can bind PFAS quite strongly, like activated carbon. And then, of course, there's a possibility that you can use thermal regeneration to regenerate this biochar, then decompose the PFAS that you sorbed. And then you create this very nice circle and a potentially very interesting value chain here for waste handling companies and wastewater treatment plants. So we start with the first part, the fate of PFAS in pyrolysis. For this, we used pyrolysis technology from Biochar, or Biochar pyrolysis technology from this French company called TIA. It's a medium-scale plant that has this electrically heated spiritool, which is a screw. So you drop the feedstock in here and it's moved along the screw and heated, comes out as biochar on the other end, and the syngas exits here in the middle. And this screw that we used could go up to about 800 degrees. And the syngas is condensed here in this condenser on the outside, removing pyrolysis oil before the syngas is combusted in a fairly simple burner with a small amount of propane at temperatures around, well, a bit above 800 degrees Celsius. Now, how did we go about exploring the fate of PFAS in this system? Well, we knew that we needed to check different feedstocks because there could be matrix-related differences. So we took two digested sewage sludges from different types of anaerobic digestions. Then we took one digested and limed sewage sludge and one dewatered sewage sludge. So non-digested, and they're all from different wastewater treatment plants. Then we pyrolyzed them at temperatures between 500 and 800 degrees. And we started at 500 because we knew already that we will need a fairly high temperature to break down these forever chemicals. Then we tried to establish a mass balance by looking at the amount going in through the feedstock, and then what remains in the biochar that comes out, and how much is emitted through the flue gas. Unfortunately, we were not able to look at the pyrolysis oil. There are no standard methods for this at the moment, and it was a bit out of the scope of our study to develop something like that. To sample the flue gas, we used this low volume air sample. It's positioned here elegantly on this shoddy plastic chair. It draws exhaust from the chimney and across this filter cartridge. We have a filter at the top to collect the particles, and then a very strong sorbing XAD resin inside that collects contaminants in the gas phase. So we have particle bound and gas phase PFAS. We also looked at a quite wide range of PFAS compounds. And this is because we know there could be a lot of them, or a lot of different ones in the sludge. And also because we knew already that they are quite persistent, but that they also, they can be transformed in thermal treatment and in natural processes. So you have precursor compounds, for example, that are transformed into more common PFAS when you heat them, for example. So the groups we looked at were these. So for you non-chemists out there, don't freak out. This is this is not as complicated as it looks. These are all quite similar, like the carboxylic acid PFAS. They have a carboxylic acid group and then a tail. And then you have the sulfonic acids, which have a sulfonic acid head. And we have the ultra short chain carboxylic acid and sulfonates, which have less than 4 of these CF groups. Then we have some fluorotelomere sulfonates, which have 2 carbons that are not saturated. And then we have some fluorosulfonamides that have an amide group here attached to the sulfonic head. And then there are some miscellaneous PFAS that are mostly substitutes for PFOA and PFAS because the reuse has been restricted. And this has been a bit of the issue with PFAS. Like we find out that some of these are toxic, and then chemical companies shift their production to something which is a little bit different, not exactly the same, but has pretty much the same properties. Then we have to start all over again to prove that they are a danger to the environment and people. And then we have some precursors. And by precursors, I mean a structure that through some kind of environmental degradation pathway can be turned into a more common PFAS, like this one, which is cleaved here, and then you have 2 fluorotelomere alcohols. Yes, so we went pretty wide. Okay, so finally some results. Our sewage sludges do contain a lot of PFAS. Our 4 feedstocks had concentrations that ranged from 56 to 441 nanograms per gram. And that is the same as micrograms per kilo dry weight. Yes. And this is quite a lot. And but these concentrations are similar to what is found all over the world. So it is actually no big surprise, unfortunately. And even though we found a lot of different ones here, as you can see, it all makes sense that they are there, because when we consider potential sources like firefighting foam, surface coatings, cosmetics, detergents, and the type of PFAS that end up in our food through, for example, sewage sludge, fertilizer application to agricultural soils, then it all makes sense that they are present in the sludge. So maybe more interestingly, we move on to the biochars, and we did find residual PFAS in the biochars, even at fairly high temperatures, like 750 degrees. But the concentrations are a lot lower than in the feedstocks. So there's about 1, 2, 3 orders of magnitude concentration drop. That's good. And also there are a lot fewer congeners. So there are only certain PFAS that survive this thermal treatment. This we could see because about 97% of what we found here were long chain PFAS. Now, when we compare the amount that was in the feedstock to the amount we have in the biochar, we can calculate these removal efficiencies, which is just the biochar concentration divided by the feedstock multiplied by the yield. And looking at removal efficiencies for all our samples, the 4 different feedstocks at all the temperatures, we can see that it was above 98% removal across all feedstocks and temperatures. And that's really, really good. There might be a temperature effect here. Removal efficiencies are slightly better at higher temperatures. There might also be a retention time effect, because these samples were run at 40 minutes compared to 20 minutes retention time in the pyrolysis reactor, not by design, unfortunately, but due to some technical issues we had, but kind of indicates that retention time matters. But what we do know is that there's a congener effect. So it matters what type of PFAS you have in the original feedstock. As I said, we found mainly long chain PFAS in the biochars, and other studies, one other study shows that long chains are more hard to break down than the short chains. And also these sulfonic acid PFAS are the hardest to break down. And it makes a whole lot of sense when we consider that this digested sludge feedstock had quite a lot of the PFAS, the sulfonic acid congener, long chain, which is the reason why the removal efficiency is slightly lower. Okay, on to the emissions. PFAS do survive the pyrolysis chamber and combustion. And we calculated through this carbon balance approach that we release 3 to 260 micrograms per ton of biochar produced. And what we emit is mainly these short chain PFAS. So we have longer chains that are broken down to these shorter ones, and they go out with the flue gas. But they are mainly associated with the particles, because between 80 and 100% of all the PFAS we found in the emissions were in this particle phase. The emissions also increase with temperature, about 50 micrograms per 100-degree increase. And this makes sense when we consider that we probably decompose more when we increase the temperature. And also we create more syngas when we pyrolyze at a higher temperature. Carbon yield goes down, as you all know. But what you wonder now is probably, is this a lot? Like, how much is 200 something micrograms per ton? Well, putting it into a bit more of a perspective, look at the mass balance, we can see that 98% is decomposed or lost, not something we could trace with the analysis that we did. Less than 2% remains in the biochar and less than 0.1% is emitted. And this is really great news for pyrolysis as a waste handling treatment for sewage sludge. To get even more numbers here, if we assume that we run our Biochar unit at full capacity for about a year, so allowing some days for maintenance and holidays, we pyrolyze 19 tons of this most contaminated digested sludge. At 500 degrees, we would have 26 milligrams in the biochar, 0.05 milligrams emitted, and 8,218 milligrams decomposed. Increasing to 700, we remove everything from the biochar. We emit a bit more, 0.76 milligrams, and we decompose slightly more. So this looks really good. But of course, there are some limitations. There always are. We don't really know everything about potential degradation products. So we only quantified what the exact compounds we were looking for. But there are some laboratory studies that show that these PFAS compounds degrade in thermal treatments through chain incision. So this chain is cut somewhere, either at the functional head or somewhere along the chain, and we get shorter fragments. And we don't only get the actual PFAS compounds we were looking for, and we also get some perfluoroalkanes, perfluoroalkenes, and perfluoroalkylaldehydes, and others. And it was out of the scope of our study to check for these type of compounds. That requires fairly advanced monitoring of the emissions. And this will also likely be very different in a full-scale plant. These results are from laboratory studies where you have pyrolysis only in controlled conditions. In full-scale units, we of course have pyrolysis and combustion, so kind of two thermal treatments. Okay, so that kind of confirms the first part. It does work, even though there are some uncertainties. Then on to the next part. Can we use sewage sludge to bind PFAS? Can we take the sewage sludge biochar we now know is free of PFAS to bind further PFAS in, for example, wastewater treatment? So to check this, we did some sorption tests. Took 2 of our sewage sludge biochars, one digested and one dewatered based, and compared it to one clean wood chips biochar. They were all made at 700. And we took this high temperature version because we already knew that to make good sorbents for organic contaminants, you need biochars produced at high temperatures. And then we did batch sorption tests with 6 different perfluorinated carboxylic acids with different tail length. And the way we do this is we take a bottle of water, and we add a little bit of the biochar, or a known amount, and then we spike with different concentrations of these PFAS. Then we shake bottles for 14 days, filter and analyze the water. And then we know by the difference of how much we put into the water at the start and how much remains in the water phase, we know how much is sorbed to the biochar. And then we can use this Freundlich sorption isotherm model to fit the data. And what we are after is this KF value, which is the Freundlich partitioning coefficient. It tells us how strongly a certain sorbent will bind a contaminant, independent of concentration. So then we plot the water concentration against the concentration in the biochar, do a log transformation, and we get this KF value from the slope. Then we can compare the KF value with other studies as well. And we look at some data here for PFOA. And to our great surprise, we found out that our sludge biochars actually had higher KF values than the wood biochar. This was very surprising. It had been established knowledge that you probably need woody materials to make good sorbents for your organic contaminants, because you want biochar with a high porosity and high aromaticity. And if we compare to some KF values from the literature, we actually see that the KF values for our sludge biochars are similar to that of commercially produced activated carbons. So this is awesome, really great news. And but before I conclude, I just wanted to try to explain why the sludge biochars are better than the wood biochar, because this kind of we're a bit puzzled by this. Because if you look at the internal surface area and the pore volumes of the wood biochar compared to the sludges, we can see that with CO2 absorption, it's almost 6 times higher in the wood biochar and 3 times higher with N2 absorption. So much higher internal surface area and higher pore volume, which is usually a key factor that decides sorption of organic contaminants. But if we look more closely at the distribution of these pores, we can see that the wood chips biochar mainly has its surface area and pore volume in this micropore range. So below 1 nanometer or even less. And the sludge biochars, they mainly have mesopore structures. And the thing is that these PFAS molecules are actually quite big. So it seems almost too simple, but the case is probably that they physically just don't fit into the micropores of that wood biochar. All right, to conclude, PFAS is removed from sludge when it's transformed to biochar if we have a high enough temperature and a long enough retention time. Some PFAS are harder to degrade though, so we need to adapt conditions to specific feedstocks. Also, the majority of PFAS are decomposed, but we do not know if some of them are trapped in the pyrolysis oil. If the pyrolysis oil is used for some kind of combustion process, we will, however, get some kind of extra thermal treatment though, so I'm not super worried about this part. A small fraction is emitted, but there are some uncertainties related to the presence of degradation products, so we might need some flue gas cleaning. Good news here is that the emissions were mainly particle-based, so they will probably be easy to lower with some kind of particle scrubber. Also, we can use sewage sludge biochars as powerful sorbents for PFAS, as long as we have a suitable pore volume properties. Okay, before I finish, I just want to acknowledge the 2 projects funded by the Norwegian Research Council, and also the whole group of people that have been involved in different parts of this process, either with the pyrolysis equipment or the lab work or administration and funding acquisition. So thank you for listening.
Thank you very much for your presentation, Erlend. You please write your question in the chat because we are a bit over time. So this will speed up the process. So you got one question from Brooke. Do you think the amount of PFAS emitted would be reduced if the syngas was also processed through a thermal oxidizer?
Yes, it's possible. We do know that retention time matters. So we have sequential steps with thermal treatments. Elonging the thermal treatment, it will probably increase the efficiency of the degradation. And there are some laboratory studies that has shown that it doesn't really matter if it's pyrolysis or combustion, as long as the temperature is high enough, like you will degrade these compounds. So yeah, that could work.
I jump on the retention time. So we saw a small influence of the retention time, or a possible small influence. Did you do any tests of going down with residence time? So is it a step change, or is it a only incremental change? If you double the residence time, you remove 0.5 percentage more or so? Do you have any information on that?
No, unfortunately not. It was fairly complex with all the feedstocks and the temperatures, and so we were or the plan was only to use one retention time. So this 40 minutes was just something that happened because we had to adjust the speed of the screw to adapt to a tricky feedstock. So we don't really know, but some other studies suggest that retention time matters also. Of course, in the perfect atmosphere, it doesn't really matter, I guess, in terms of carbon balance, but in degrading these PFAS, it might have an effect. And also it could be, if retention time is an important factor, as we believe, you could maybe lower the pyrolysis temperature and increase the retention time to get same type of efficiency.
And so please write your questions in the chat, so you will have another minute to write them. Another question from myself about your degradation products. Do you have any information on, or does anyone know if potential degradation products that we know of are actually more or less toxic?
Yes, so this is one of the unknowns also. We don't know too much about the properties of a lot of different PFAS. We know the common properties they all share. This functional group, though, like this head group, is quite important. So when you have perfluoroalkanes and alkenes, they will probably not be as mobile. They'll be more hydrophobic, so they probably won't dissolve in water as easily, and they won't have these surfactant type of properties that the other regular PFAS molecules have. But it is still an unknown, like the kind of impact such compounds would have. Like we don't really know for a lot of different PFAS as well, how toxic they are and what kind of impact they have.
I guess that's the downside of limiting certain ones and then getting new ones. So you get so we have three last questions for you. First one from Joel is: did you consider total organic fluorine analysis?
Yeah, no, we did consider it, but we did not have a suitable partner at the moment, and we couldn't fit it into our budget. But yeah, that would probably be a good idea. For example, a first look at the pyrolysis oil. But it's tricky to establish mass balances for these full-scale systems. You know, it's for any kind of compound.
Yeah, especially concentrations are not that high.
Mm.
So Sylvia was asking, does your research indicate that we must be careful in pyrolysing sludges to avoid risk of releasing some of these compounds in the exhaust gas?
Not really, or of course emissions matter, and even though they're quite low, it is could be significant. The effect of flue gas cleaning should be investigated, because this unit did not have any type of flue gas cleaning. So if you have a particle scrubber, it could have 80 to 90% efficiency in removing particles, which would remove most of the emissions we have here. They're so associated with particles.
Okay, and then I have a really good last question for you. From
alternatives we have for unfilling leads to emissions. Excuse me.
So you were lost for a second.
Oh, Yeah, I just wanted to say that, yeah, all types of treatments lead to emissions. Landfilling leads to emissions because they're so mobile. Waste incineration will also have some of the same issues as we're discussing here. And of course, using the sewage sludge directly as fertilizer is not such a good idea. So yeah, there will be emissions no matter what you do with PFAS containing waste. It's a cyclical problem. So the fact that we're actually removing 98% of known PFAS compounds here should be considered a big win. But I would, of course, suggest that we look into efficiency of flue gas cleaning and also try to measure the presence of degradation products also in full-scale units, even though it's difficult. You need fairly advanced equipment then.
So perfect last question from Ingrid. So great work. Is it planned to use this at a larger scale in Norway? Do you have any plans do you know of any plans that this will become standard treatment?
Yes, so our industrial partners, they have big plans to use pyrolysis for waste treatment. So it's at a fairly early stage right now, but there are multiple actors that are investing in pyrolysis equipment. So the results from this project will be probably used by the Norwegian government to try to establish some a baseline or some used as data to make emission thresholds, for example. And yeah, so yeah, it's still in an early phase, but it might become a big thing if it keeps on moving the way it is now.
And I guess if you keep researching it, so you will lower the barriers to commercialization, hopefully.
Of course, but our industrial partners are very serious about this, and one of the research projects is this industry research collaboration and research and innovation project. So, so they are developing the technology while we're writing the papers.